All right, welcome back, everybody. Today we will continue working on the spatial transcriptomics that we have been slowly working our way through with the human cortex samples, human frontal cortex samples. So if you guys are finding this helpful, please let me know. It does let me kind of know whether or not I should keep working on this kind of stuff or if I should go back to some of the original videos that I was working on, which are more focused on using bioinformatics tools like alignment, gene set enrichment analysis, pathway enrichment, that sort of thing. So if we jump into our studio, we can see here that I have a script open where I have loaded in some of the libraries. If you guys want to follow along, I would suggest installing as root these different libraries here. So we need libproj, libgdal, libudunit2, and then libgeos. And what these are going to do is it's going to allow us to use the package spdep, which is going to help us with some of this stuff that we're talking about today. So here is the GitHub page. I will go ahead and upload the script into the scripts folder. So if you guys have missed the previous one, feel free to kind of start at the beginning and follow all the way through. So today we're working on number 7, which is going to be spatial autocorrelation of two different genes. We're going to look for ligand-receptor pair relationship spatially to see whether or not there's any cell-to-cell communication happening. The database that we're going to use for the ligand-receptor pair gene ligand-receptor gene pairs is in this CellTalkDB. So if we go back to this homepage here, they do have a paper. Feel free to go ahead and read that. What we're going to use is the human ligand-receptor pair gene list. Let me adjust this here quick. And in order to use that, what you can do is just click on it and then right-click, select copy link. And inside of our studio, to get that loaded up, you can actually paste in the read RDS, since it's an RDS file, and specify that it's a URL. And what that's going to do is it's going to download and import it right into R, so you don't have to save it to your machine. Like I had mentioned, we're going to use this spdep package. I was having some issues with this before, but what I had noticed was that you need to download it from their GitHub. Because on GitHub, they actually have the function that we're going to be using, which is Moran _BV or bivariate. We're going to look for two genes and see whether or not they're spatially related. The expression is spatially related. So this isn't a point pattern anymore. We're looking at continuous data. So is the regions on the sample with high expression of one gene also the regions of the sample that have high expression of the other gene? And that's going to be sort of a proxy looking at the gene expression for whether or not there is protein for the ligand and protein for the receptor that potentially are talking to each other. All right, so let's go back over here. So we have all of our libraries loaded in. Awesome. The next thing that we can do is we can load in our gene expression data and then our metadata. And just like last time, what we have to do is we have to create that extra column that's a key between the slide that we're working on and the field of view. So in this case, we might not have multiple slides. This might be all the fields of view. You can actually look here. All the fields of view from a single slide. But if you have many samples, what you'd want to do is, yes, they're all from the same slide, is make sure that there's that key there. So this is just a good habit to get into. So if you have multiple slides, you're not confusing the fields of view on one with the fields of view on the other one. Next, we'll go ahead and we'll load in the gene sets just like we had done in 6.1, where we're looking at the human hallmark gene sets. We're going to specify category as H for hallmark, and then the species is Homo sapiens. After that, we have to split our gene set list. So if we look at all gene sets, it's just a long table. We're going to split it on the actual gene set and create a list that has the vectors of all of the genes that correspond to that gene set. After that, we can load in that ligand-receptor pair database that we had found with the CellTalkDB. Once we have that done, we're going to stick to a single gene set today. So from the previous 6.2, we had looked at if there was spatial clustering of the cells that have high gene set scores for any particular gene set. So here I stuck with the epithelial mesenchymal transition gene set, but if there are others for your data set, that stand out in that heat map, feel free to substitute this in here for what is in your, this guy, for what is in here. So if you have a different one that's not the epithelial mesenchymal transition that's really standing out as having significant spatial correlation or significant spatial enrichment, you can replace our epithelial mesenchymal transition with whatever that happens to be for your data. And what we're doing here is we're just pulling out the genes that are in the ligand-receptor database, gene pair database, that are also in our CosMx SMI data. So in order to do ligand-receptor pair analysis, we have to make sure that the ligand and the receptor are both in our data set. And then next here, we're just filtering out the ligand-receptor pairs from that data to include only those that have a ligand and a receptor. So we're kind of filtering to get only the genes that are part of the epithelial mesenchymal transition in our data, and then the ligand receptor pair genes for our data as well. So we're kind of double filtering it to make sure we're only getting the genes that work. So next we actually move on to the spatial autocorrelation using Moran's eye. So like I had mentioned, we're going to look at the expression of our ligand, and then we're going to look at the spatially lagged, which is basically taking the average of cells that are the nearest neighbor to our cell of interest, the average of those. And we're going to see whether or not the ligand expression, where it's high, has high lagged, spatially lagged expression of the receptor. And what this can tell us in proxy sort of is that we have high expression of the ligand and the receptor. Perhaps we have high expression of the ligand and receptor proteins that the cells are using to communicate with each other. And the nearest neighbor is just telling how far out from our anchor cell, right? So if we have a cell here and then we have surrounding, 10 cells, we only want to look at the cells that are really close by if we're looking at the communication between cells. It doesn't make sense to look at cells that are, the 50 nearest cells, for example. It doesn't make sense to look at something like that. So we're going to stick really close. The 3 closest cells to our cell of interest is what we're going to use to spatially lag our data. And then the fields of view, this is just getting that vector that we're commonly using throughout all of these different scripts. And it's going to allow us here in our lapply to loop through each one of our fields of view. And something to keep in mind here, these fields of view are all from the same slide. So let me see if I can find this, this study somewhere. But when we're using, so the moral of the story is when we're using a bunch of samples from the same slide, even if they're from the same sample, remember the different fields of view on a slide can be from one sample, or we can have something like a tissue microarray where we have many different samples that are gridded out. But if we have even a single tissue and we're taking a bunch of fields of view from that, we want to treat them all separately because tissue heterogeneity may cause structure to start popping out if we look at everything. And different areas of the sample might have different characteristics that mute each other. So here, we're going to keep the fields of view separate. That's kind of the main plug that I'm making there. So if we look at how we're going to calculate the Moran's eye, we're using inside that SPDEP package that we have to get from GitHub. What we're going to do is we're going to filter down the metadata first to include only the cells that are from a particular field of view. And then what we're going to do is we're going to select the genes, the gene expression for only those cells. So we don't need the gene expression for all 200 or 391 other fields of view in this sample. We just want to look at one at a time. Then we're going to create this SPDF, which is a SF object with our gene expression table. So we'll have what this field of view expression data frame is going to have. It's going to have our cell ID, the X and the Y locations, and then all of the genes that are associated with those cells. And we're going to tell this ST to SF when we're creating our SF object that the coordinates of each cell are in x underscore FOV underscore PX, so the pixels for the x location, and then y underscore FOV underscore PX, the pixels for the y location. That's the center of our cell. And then what we can do is we can keep passing that through. Next, we'll look at the k nearest neighbors, and we're going to set it to that nearest neighbor that we had set above. So if you want to look at a little bit broader nearest neighbors, what you can do is just change this to something like 6 or 10, and then you can look at the nearest neighbors in a little bit bigger area. But you have to remember, if you're looking at cell-to-cell communication, you can't go too far out, because if the cell is on the other side of the field of view, it just doesn't make much sense. But we're going to look at the nearest neighbors for each of our cells and the 3 nearest neighbors. The next thing we have to do is convert that to a neighbor object. So k nearest neighbors to neighbor, a neighbor list. And then we're going to create a list, a weighted list. So basically each cell's nearest neighbor is going to have a weight associated with it. And here we're using binary, which just means that if it is a nearest neighbor, anything that's included in this case, it's going to get a 1. And all of the other cells that are not nearest neighbors are getting a 0. They have no weight. Yeah, you can also use W, so they recommend row standardized, but that would just be the number of cells that are nearest neighbors divided by, or 1 divided by the number of cells that are nearest neighbors. It's just a little bit less of a weight. But otherwise, it seems to work either way. You'll have potentially lower Moran's I values using the W. But next, what we're going to do is we're going to go inside of another lapply, and we're going to go through each ligand receptor pair that we had found in our data set that has genes from the EMT hallmark gene set. So each ligand receptor pair we're going to pass in. And what we're going to do is we're going to basically look in our EMT pairs, so the big data frame that we had looked before. We're going to find the ligand receptor pair that is part of that row back here. So we're passing all of this in. We're going to basically filter down to whatever row that is, and then pull out either the ligand, if we want to look at the ligand, or the receptor, if we're going to look at the receptor. And that's what these 2 rows are doing. We're basically pulling apart in a way that will allow us to make sure that the ligand receptor pairs are in our data. And we're splitting this apart. So we're going to grab this, and we're going to grab the ITGB1. If you know that each one of these are in your data, like they should be here, you can just split this on the underscore and use the first part as the ligand and the second part as our receptor. But that's all we're doing there. Next, we're going to actually calculate our Moran's I bivariate. So we're going to look at our ligand genes, and then we're going to lag our receptor gene expression. And then we have to pass in our weights that we're going to use for those cells. And then what we're doing here is using this NSYM in order to create a sort of bootstrap in order to then later pass it into this in order to find out our confidence intervals. So if your computer is a little bit slower, you can use 100. You'll just have potentially broader confidence intervals when going through there. But then here, we're going to use the boot.ci confidence interval in order to create the confidence intervals for our observed Moran's I value. Finally, we're going to return for each one of our ligand receptor pairs the original value, which is in this T0. We're going to return the actual ligand receptor pair, which is this guy here in the first column. And then we're going to return the low confidence interval, 95%, and then the high confidence interval marker for the 95%. Once we have all of those values for all of the ligand receptor pairs, we're going to bind them together. So for each field of view, we're going to get a data frame that is of length ligand receptor pairs. So we have 68 here. For each field of view, we're going to get 68 row data frame. And then we're just going to use a bunch of cores and try to get it done really, really fast. So if we go ahead and look at that, We can see here we have an entry for each one of our ligand for each one of our fields of view. Then we have a list, so it's a data frame, and it has 68 rows in it, one for each of the ligand receptor pairs that are found in our data. Awesome. So once that's done, I recommend saving it because it does take a really long time, and it just makes it a lot easier in the event that something happens. Then you can import it again. You don't have to go back through and rerun everything because that can take a really long time. What we're going to do is we're going to bind it all together. So instead of having a list where each element is a field of view, we did tack on the field of view down here. So now we're able to row bind the rows for each one of those elements and get a really big data frame. So if we look here, we have the first 5. I shouldn't say the first 5. We have the 5 elements that are in our Moran's list, 1, 2, 3, 4, 5, and then each field of view will have 68 rows. So in total, we have 26,656 rows. From there, I had arranged it descending. That way we can see the highest Moran's I value. And this is going to take, for your particular data set, you're going to have to explore and figure out whether or not the output makes sense. So in this particular example here, if we look at the head of this data frame, we can see that for slide 1, field of view 225, we have a Moran's I value of, a bivariate Moran's I value of 0.93 for SPP1 and ITG beta 3. Okay, if we go ahead and actually look at this though, let's do that, and then we can plot. We can see, okay, there is some open space here that might make this estimate a little unreliable. But let's go ahead and look at the ligand expression. Okay, we have one really high here. Otherwise, it looks like most of it is very low. Maybe some moderate up here since it is normalized. Maybe one down here. If you look at the receptor expression, we can also see that it's a couple genes and the region just happens to overlap a little bit. But have to take it with some thinking through, critical thinking of if this actually makes sense. So let's go ahead and we can look at another one here. So let's look at slide 1, FOV 217. There's only in this 73 cells or something like that. 73, 68 observations. I think it's 68 cells. But anyway, so let's look at this collagen 7 alpha 1. Let's see if I can find how to do this with my brain. Okay, and then we'll look at the integrin beta 1 down here. All right, so let's look at this FOV. Okay, so our cells are a little bit more together. So let's look at the ligand expression. And we got one cell again with high expression and the rest seem to be very low. There is maybe another one moderate in here. It's not a great scale. One down here. And then let's look at this guy. Oh, so we can see we have another cell. It's not, is it the nearest neighbor? It might be the nearest neighbor. So that might pull a lot of weight. But anyway, so now we have this field of view versus Moran's I value for a ligand receptor pair. So what we can do now, if we had different treatments of our cells or our different samples, fields of view, we would be able to go ahead and compare perhaps the fields of view that are from treatment. So some, I don't know, chemotherapy, for example, have much greater Moran's I of a ligand receptor pair than, say, those that are treatment naive. They have not received treatment or that chemotherapy for that same ligand receptor pair. So there's some statistics that can be done now with this output for groups if we had different sample groups. But yeah, so that is the cell communication that you would have to dive into on your own to see whether or not, like I said, it makes any sense that you are seeing this spatial autocorrelation in your samples. You probably want to find some genes that have more cells with higher expression. That way you're not looking at a single cell with high expression and a single cell with a high expression for the ligand and the receptor respectively. But yeah, so now downstream comparisons for your samples should be able to be done. Let me know if you have any questions, and thank you guys for watching. Bye.